So the last part of chapter 5 is about a really key protein in our body in the world of biology, and those are enzymes. So enzymes are proteins, and what they do is they speed up chemical reactions. They are biological catalysts. You might say that they drive chemical reactions. The other thing that we could think of in functional terms is that enzymes digest our food because they are breaking down those macromolecules. And breaking down those macromolecules is a chemical process. And so basically what we're talking about with enzymes is what they do is they make things easier to happen. So they speed up the rate of a chemical reaction. They make it happen easier by lowering the energy barrier, or the energy of activation. That's what E of A stands for. And they're not used up in the process. So if we were to talk about this, there is a lot of energy in biological molecules, such as carbohydrates. Digesting them would be an exergonic reaction, right? But in order to get from my reactant to my product, sometimes you have to break some bonds in order to make the new bonds. And so sometimes there's this idea of a toll to pay. You know, they say sometimes you have to spend money to make money. The same thing is true with chemical reactions. There is a bit of a barrier or an energy of activation. This is the toll you have to pay. This is the money you have to spend in order to make the money, right? And so what happens with an enzyme is that it makes it easier. It's going to lower that. So rather than this being a tall bump, it's going to say, oh, well, there's an easier way for it to happen. And when it's easier, the chemical reaction can happen more, or we say it happens faster. And so that's why it speeds up the rate of a chemical reaction. I love a graph. I couldn't help myself but draw a little scribbly one there. There's a much prettier one here. So this is a chemical reaction. It would be an exergonic reaction because we've got a lot of energy in the things we start with and a little energy there. Generally, we're going to have this amount of energy coming out, right? If we were to do it without an enzyme, so this is the chemical reaction without an enzyme, look at this big hill it has to climb over. This is the barrier. This is the money you have to spend in order to make that money. It's the toll you have to pay. And so with the enzyme, you can see that it makes things easier. There's a much lower barrier. There's a much lower cost to this chemical reaction. And that's really on a chemical level what happens with an enzyme. So hopefully by understanding that energy in the cell, we're getting closer to understanding what an enzyme actually does. There are so many enzymes out there. It's important to understand these. So enzymes, being proteins, form and function is everything. If an enzyme has the wrong shape, it's not going to be able to do its job. Each enzyme actually has a very particular shape and it fits the substrate. So this target molecule literally fits in like a lock and key idea. So it geometrically fits into the enzyme. If you were to talk about a specific example for these, the milk sugar lactose fits the enzyme in your small intestine of lactase. If you're lactose intolerant, it might be that the lactase isn't the right shape. So it's a result of the shape that the enzyme has an active site. It's this active site where the substrate comes in and bonds. As it's held in the active site, that's where the chemistry is altered to make the product. So we had a substrate go in and a product come out. If we were to shift the conditions, we could denature the enzyme. This happens a lot if you cook things. And so if you have a high temperature or change the pH or change the salt level. We talked about this back when we did proteins back in chapter 3. So in terms of now understanding this, I love this picture. So this is not lactose and lactase, but it's the enzyme sucrase. So still a protein, and you can see this shape up here is such that it actually fits the substrate. You might talk about this as the reactant of a chemical reaction. So it literally fits into the active site. Look at this one more time. We've got water coming in, so that would be hydrolysis. And then we have two monosaccharides as the products. And the other key thing, and it said this in our notes, is that the enzyme is not used up in the process. It can be ready to go again for that next disaccharide substrate to come into it. Chemical reactions and enzymes and sort of like how things are regulated is really important. So if there's a chemical that interferes with enzyme activity, we call that an inhibitor. So many drugs actually work as inhibitors if they inhibit the actions of things that we don't want to have happen. So they might inhibit cells or chemical reactions in your cells that cause swelling. So ibuprofen works like that. It stops chemical reactions and decreases the swelling in your body. Or if a drug acts as an inhibitor on another cell, like a bacteria that is invading you, then it's going to kill the bacteria. That's why you don't take antibiotics for a virus. You're trying to kill a cell that has actually come into you. On the other side, there are some pesticides and poisons that also act as enzyme inhibitors. One example of a really bad one would be antifreeze. It's classic. Dogs love the taste of it, but it acts as an enzyme inhibitor and is not good for your dog. Alcohol also is an intoxicant, and it works on a cellular level, inhibiting chemical enzymes. And so that's some examples. Outside of antibiotics, you could also have cancer drugs, anti-cancer drugs, right? Where it interferes with DNA production and therefore cell division. So we're going to talk more about cell division and cancer on in chapter 8, but these are just examples in understanding inhibition, what is a good thing. Now we can have competitive inhibition or we could have non-competitive inhibition. Competitive inhibition is when a molecule is really close to the shape of the substrate, and it's going to basically get in there and block the active site. Non-competitive inhibition means that a molecule budges into the enzyme in a place other than the active site and alters the shape of the protein, which makes it non-functional. So I love some pictures of these. I find them to be really useful in sorting my understanding of this stuff out. And so at the top there you have the normal action of enzyme and on its substrate. And in competitive inhibition, this might be an antifreeze molecule blocking the active site so the actual substrate can't get in. Or down here, this would be a non-competitive inhibitor, and it budges in and it changes the shape so that our substrate can't get into my active site. So in this way, it is important to regulate chemical reactions, and most of our chemical reactions in cells have an enzyme. The last little bit of the notes here is really just a preview to chapter 6. I would say that chapter 6 is a lot. You can take a look at these as you're doing your pre-readings maybe for chapter 6 and 7, because when you open your book for chapter 6 and 7, it's a lot. And so I put them in here just to give you like everything kind of condensed into just a few words. So I'll remind you that they're here. I'm not going to talk through them in the video. But yeah, chapter 6 will be up next.